Okie dokie. Let's give this a shot. Today is the blood lecture, first part of the cardiovascular system that we'll be looking at. And we all know that blood is a connective tissue. Remember from histology. And it has a fluid extracellular matrix. Or ECM. The functions of blood include: it connects the internal and the external environments. So we're basically talking about air to the organs inside your body. And it transports oxygen. Delivers nutrients. Oh. It also transports hormones and wastes, metabolic wastes primarily. It also functions to maintain interstitial fluids, which is the basically the fluid that's between cells in your tissues. It also distributes heat throughout the body. Distributes heat. And one more down here. It is part of the immune system, so it defends against diseases. Defense from disease. Okay. This is gonna be tough. Ooh. Well, you guys can rewind, right? So here you go. Next slide. The blood is composed of what are called the formed elements. And plasma. The formed elements are the cells, and the plasma is, like I said, is the extracellular matrix. We'll talk more about what that actually is. The formed elements include the red blood cells. I usually just shorten that as RBC, red blood cells. Also the white blood cells, which are important for the immune system. Blood cells, or the WBC, and we'll talk more about those. There's a bunch of them. And then what are basically fragments of cells, the platelets. And these are important in helping stopping bleeding. So the red blood cells, if you were to take a milliliter, let's say, of blood, About 45% of that, roughly, is going to be red blood cells. The white blood cells make up about 1% of it. And roughly platelets would make up like almost nothing. Roughly half of the blood, so these are the formed elements, is plasma. It's about half of blood. This is actually a little bit low. It's closer to like 48, 49, almost 50%. And it is made up primarily of water. About I think it's 92%. It's also got some proteins that are dissolved in it, amino acids, and different biomolecules. Let's just call them biomolecules. Carbs, lipids, etc. It also contains hormones and electrolytes, which is the fancy term for basically ions. We've talked about calcium and sodium and all those. So the ions or the electrolytes are, and because other electrolytes as well, are travel around through the blood. And we talked about that. We talked about hypo and hypercalcemia, and I talked about if your circulating calcium gets low, then you pull it out of the bones. This is what I'm talking about. The calcium is flowing through your blood, and if your body detects that it's too low, it starts dissolving it so the circulating ions start to go up again. And you also have different wastes that are produced by the cells. How much blood do we have in our body? I wish I could, it's like interactive. Typical adult is about 5 liters of blood. Liters. Okay. Red blood cells. RBCs. The fancy sciencey word for that, they are called erythrocytes. And they are described as being biconcave discs. That's not a very good drawing, but they're kind of that shape-ish. They lack a nuclei, so biconcave discs. They lack a nucleus. They used to have one, but they kicked it out. It makes more room for hemoglobin, which is kind of the whole point of the red blood cell, is to carry oxygen. So it's just going to live for a couple of weeks, I think, and then it dies. So it doesn't really need a nucleus. It's not going to divide, which is the whole point of the nucleus, is to allow it to undergo mitosis. They lack mitochondria. And they're filled about 1/3 of it with hemoglobin. The shorthand for that is HB, or you might see it as HGB. They call this the oxygen carrying, they call it a pigment. And when you have hemoglobin with oxygen bound to it, they call it oxyhemoglobin. And hemoglobin without oxygen That's without. It's called deoxyhemoglobin. There are a few ways to analyze the blood. Like if you go to the hospital, that we're going to draw. Well, I mean, there are other tests, but one of the things they do, or a couple of things they do, is check the red blood cell count and your hemoglobin concentration. So, red blood cell counts, and this is what they say per microliter, which is. So it goes milli, micro, nano. So this is 1/1000th of a meter. This is 1/1000th of a milliliter. So it's a very, very, very small amount, like probably less than a drop. And what they do is literally they'll put it on a microscope slide. And on this slide, they've got this grid, and then you can't really see it with your naked eye, but when you look at it under a microscope, they have this grid, and they'll put blood on there, and can't even see that red. Under a microscope, they'll go through and count, say, 20 of those squares, and count how many red blood cells are in that, and then kind of like find an average for the microliter of blood that they've drawn, and then they can kind of scale that up and figure out what is kind of normal. So the red blood cell count measures the O2 carrying capacity in your blood. I'm noticing that there's a typo in the PowerPoint. I put millimeters cubed, which I don't think that's right. Another way of analyzing the blood is through what's called hematocrit. Hematocrit. And this measures the percentage of your blood that is made up of red blood cells. And this is one of the things that we would have done in the blood lab is you have these really thin, they're called capillary tubes, and you fill it up with blood and plasma, right? And then they put it into a centrifuge and spin it around super, super, super fast. And then the tube will come out with the red blood cells separated out from the plasma. And so here's like the top of the blood sample. And roughly, in normal blood, roughly half of it will be the red blood cells. And then the other half will just be kind of this yellowy, kind of straw colored, and that's the plasma. So basically what you're doing is, you can measure this, say this is, I don't know, say 3 centimeters. Oops, not meters, centimeters. And then you would measure the length of that and then the length of that, and you set up a proportion, say that this is 1.5 centimeters. So 1.5 centimeters out of the total 3 centimeters. That means the hematocrit, or this is actually shorthand, HCT, is, in this case, easy 50%, right? Because that's about half of that. That's the hematocrit. And I'm running out of room here, but number 3, another way is measuring the hemoglobin concentration in the blood. And basically what you do is you would poke your finger, put it onto this special type of it's an absorbent paper, and it's going to be like a shade of red, and then you use this chart to compare it. Like it could be slightly more orange or slightly darker red, and you can figure out from that roughly like the hemoglobin concentration within your blood. Okay. So in order to, well, when new blood is made in the red marrow of your bones, the process is called erythro, that's an R, poiesis. This is the production or formation of erythrocytes. Red blood cells, aka erythrocytes. And depending on your age, when you're a fetus, it happens in the yolk sac and the liver and spleen, and then after birth, it's in the red bone marrow. So, as adults, this occurs in the red bone marrow. Oops, it's sloppy. So we've talked about stem cells that are basically unspecialized, kind of undifferentiated cells that can turn into almost whatever they want. So the early, before they're red blood cells, there's sort of like a midway point between like a true stem cell and a red blood cell. They're called hematopoietic stem cells, and they're also called hemocytoblasts, but take your pick. I prefer this, even though it's a very odd spelling. So this is basically the starting point of red blood cells and white blood cells. And the process, if you want to look online, it's rather complicated. So just take my word for it. You're getting the easy way out on this one. But so these hematopoietic stem cells, they can go different routes to become a certain type of white blood cell or another type of white blood cell or a red blood cell. They become more specialized as they develop, right? Of course, this is all controlled by the body. It doesn't just happen automatically. And there's a hormone that regulates this. Erythropoietin. It's the hormone that controlling RBC production. It's released by the liver and the kidney. And there's a negative feedback mechanism that regulates it. So if you need blood, it starts releasing the erythropoietin, which causes the hematopoietic stem cells to turn into red blood cells. And when you have enough red blood cells, it starts shutting this thing off, like filling up the gas tank in your car. You have an empty tank, you start filling it up. As it gets more and more full, it stops filling it up, right? So negative feedback mechanism. It also responds to low oxygen level. Low T levels. So like if cyclists and a lot of like runners and stuff like that who need a lot of oxygen for doing their sport, they'll go and train up at like Lake Tahoe at elevation where the oxygen levels are really low. And what happens is your body will start to produce erythropoietin, and therefore you start making more red blood cells. So you train up there at elevation, and then when you come back down to sea level, you have more red blood cells, so you basically can carry more oxygen. And the idea is it makes you more competitive, right? This is shorthand. We've talked about this before. I think EPO is the name of the kind of street drug that people can take. It's what people use for doping in like cycling. It was like, well, it's still a huge scandal. People use that so they can ride harder, faster, and longer. Some of the problems associated with that, you got to think of the blood as fluid, and as you increase the number of red blood cells in blood, it can lead to what's called cythemia, which is basically sticky blood. Because it's thicker, it's got less plasma per volume, right? More red blood cells. And so rather than being, say, 48% red blood cells and 52% plasma, now it's maybe only like 53% red blood cells and 47% plasma, so it becomes less fluid and more sticky. And this can be a problem because obviously it could, get stuck in some of the smaller blood vessels and cause some other issues. There are different types of this, but if you have low red blood cell count, I'm sure you've heard this term, anemia, but there are different types of anemia you can have caused by not enough hemoglobin, not enough red blood cells. But that's one of them. But you can imagine what the symptoms for this would be. You'd have low energy, fatigue. You'd have pale skin. There are some things that you need in your diet that affects the production of red blood cell. Diet effects. You need vitamins B12 and folic acid. Your body doesn't produce it, so you need to get it. This is required to synthesize DNA. Synthesis. So if you don't have this, the hematopoietic stem cells are unable to divide and produce new red blood cells, so you run out of red blood cells. Or not run out, but get low. You also need iron in your diet, which is just a trace element that you, that a lot of times it'll be like in cereal. I've heard that if you took a magnet and like a Wheaties, I guess, and crushed it up and took a magnet and rubbed it around, that you could pull out the iron chunks from the cereal. Never tried it, but iron is needed in hemoglobin. The iron is actually what holds on to the oxygen in the blood, which is if you're seen like a cast iron pan. If it gets rusty, if it becomes oxidized, it turns red, right? Well, same thing with blood. If you oxidize the iron in your blood, it turns red. That's why blood's red. One of the reasons, I should say. It can be recycled from old blood cells. Be recycled from old red blood cells. But also needs to come from your diet. So red, like all cells, they have a lifespan, and the lifespan for red blood cells is 3 months, 120 days. What did I say? A couple weeks? Why was I wrong? 120 days. And then over time, they just become fragile and damaged as they move through the blood vessels, and they just eventually they get pulled in by the spleen and liver to be recycled. So in the liver and spleen, I'm pretty sure we've talked about macrophages before. Macrophages, they phagocytize bacteria or old blood cells and basically dissolve them away, like digest them. Phagocytize the old RBCs and then break them down into their components, into the iron. The inorganic part is called the heme, the iron part of it, and into the globin part, which is basically the proteins or the amino acids. Let's see, and protein subunits. So I'm going to try to keep these to about 20-25 minutes. So that's the end of the red blood cell section. So I'm going to stop there and pick up with white blood cells in the next bit of the unit, talk about plasma and stuff. Right?